Every time scientists invent a new telescope, microscope, or X-ray machine, they open a window into a part of the world we never knew existed. Not only do we learn new things, we have new questions we never even thought to ask. On the pier behind me, in the little shingled shed, is an imaging flow Cytobot. It's a new kind of instrument that photographs phytoplankton on the fly in real time. It's part of a research project that is run by Professor Colleen Mouw at the University of Rhode Island. I heard about the imaging flow Cytobot a few years ago, and at the time I thought, wow, this is totally cool. I've always thought there's biodiversity unimaginable in the ocean, even in every drop of water. And here's somebody with an instrument that can explore it. So I thought I would call up Professor Colleen Mouw and ask her to show me how this thing works. And I thought, I'll make a video about it and then you all can see it too. I'm David Gregg. Let's take a look.
The Rhode Island Natural History Survey presents videos to showcase the animals, plants, geology, and natural systems that surround us, and the people and organizations working to understand and conserve them.
So I'm Dr. Colleen Mouw. I'm an associate professor here at the Graduate School of Oceanography at the University of Rhode Island. And I'm sharing with you today an instrument that we call the Imaging Flow Cytobot. It samples phytoplankton continuously around the clock, and we've had it deployed here since 2017. So we're pumping water in from outside in the bay and then into this instrument that takes pictures of individual phytoplankton cells.
Before we go any further, this is probably a good time to talk about what phytoplankton even are. Plankton are microscopic organisms that float in water. And they come in two flavors, zooplankton, which are animals, and phytoplankton, which are plants. So microscopic one-celled algae, diatoms, and some other stuff. What's the point of studying phytoplankton? Well, phytoplankton are the primary producers of the ocean. They convert sunlight into food for all the other stuff that lives in the ocean, so they're super important.
We were bringing water in from the bay, and it first passes through a pre-filter. There's a filter on the intake of the instrument here. Yep. And so it's got a filter for 150 microns. Given the fluidics of the system, that it would clog itself if it had particles larger than that. So this pre-filter has a 400 micron filter on it to kind of get rid of the big junk. And then we have a 150 micron filter here for just the cells that are able to move through the fluidics of the system. So the water then moves into the instrument, and inside there's a camera that is taking pictures individually of the cells. There's some sheath fluid that's constantly running through the image cell, and that causes the cells to align within the cell for their individual images to be taken.
So whatever's in the stream gets basically put into line.
Yes, exactly.
And then they go one at a time through the imager.
Yep, in front of the camera, so then you end up with a nicely oriented, in focus picture of each phytoplankton cell. It's continuously flowing through, so the camera is operating very, very quickly to capture all of these images as they're moving through. What you can't see here is that as the water is moving in here, there's a syringe that pulls the sample up. So it takes about 15 milliliters of sample, and it slowly pushes that out to allow the imaging of these cells. And so for a given sample, it depends on what's in the water at any given time, we have thousands of images of phytoplankton per sample, which is equated to millions and millions of them over the time frame that we've had this instrument out here.
What happens to these thousands and thousands of pictures? Well, if you had to identify them all manually, you'd never get through them. So there's an artificial intelligence that screens the pictures and does a lot of the ID work. Then the pictures go up on a website where you can explore them.
So the blue bars across the top are weeks, going back several years, and you click on a blue bar and you get this mosaic of the phytoplankton that were found during that week. And you can click around. Different seasons produce different phytoplankton. When you click on one of the pictures in the mosaic, you get an enlargement. And you can also see the outlines or the blots that are made by the artificial intelligence as it's going through the process of IDing the plankton that are in the pictures. You can click on big ones and you can click on little ones. You can click on winter ones and summer ones. It's interesting to see how the number of images and the kinds of organisms in the images vary.
And now we're in my lab at the Coastal Institute on the Narragansett Bay campus, and I have a second one of these instruments with outside of its can, so I thought you'd be interested to see what does the inside of this instrument look like. So before you were seeing just the top of the instrument and it was sitting within a can. So that extra can is set to the side, so now you can see what's going on here. So the sample is brought into the instrument, and there's on this side of the instrument, maybe I can turn it for you. There is a syringe here. That's where the sample is pulled into, and then as it's sampling, it's very slowly pushes the sample down into a sampling cuvette that's in this area. Here's the laser.
Okay, hold everything. Laser? What laser? Actually, the imaging flow Cytobot hits each piece of schmutz that comes through with a laser, and the light that scatters off of the object can help the computer determine whether it's phytoplankton or zooplankton or some other piece of junk that isn't worth photographing.
And then the camera is on this side that's taking pictures of the cell within the flow cell that you can't see very well because of the protection due to the laser. Down here, this is where the sheath fluid is stored that is pumped through that flow cell to make sure that those cells are aligned. And of course, you see all kinds of electronics to run everything. There's a computer on board that runs the software and the data acquisition, and
So
all
the-
of the functional
so-
pieces.
So the image processing, the identification doesn't happen on board that.
No, so this instrument, this cable that you see here, this big blue cable, is connected to a computer down on the dock, and that computer then talks to our database to collect all of the information from the instrument. Yeah, so I'm really interested in pushing remote sensing beyond just chlorophyll concentration or how much phytoplankton is there to move towards how much can we discern about the composition that's present. So there's been some of my past work and some of my colleagues have done some of this work from multispectral satellite images where we're able to pull out broad size groups of phytoplankton from satellites because different size cells have different absorption magnitudes. So the way that I can explain the size impact really quickly. So a very large cell has a lot of cellular material between the cell wall and the chloroplast. And so that mutes the absorption efficiency of that cell. When you have a very small cell, that cell wall is right next to the chloroplast. So they're very efficient absorbers, and you see the difference in the magnitude in the absorption spectra. And essentially that translates to what we're able to differentiate from satellite.
Right.
My big motivation of wanting to sample the phytoplankton in this way is to connect to other segments of my research where I'm looking at optical properties and how they relate to what we're able to see from a satellite. So by collecting the phytoplankton composition alongside a whole suite of optical instruments that measure the absorption and the scattering of different optical components, we can then look at how those things, how the phytoplankton composition is shifting the absorption and backscattering spectra. And the satellite that's looking at the ocean is measuring remotely sensed reflectance, which is a function of that absorption and scattering that happens from the water itself, the phytoplankton, and the other optically active constituents in the water.
Natural History Survey videos are made possible through the generous contributions of members and friends. Want to help us do more environmental science and conservation? Hit the like button, share our videos with your circle, subscribe, or make a financial contribution on our website, ranhs.org, or through Patreon. Thanks, and see you out there.